Hope everybody's doing well. Sorry, it's good afternoon. So, we will be having a session today about biological molecules. We're going to wrap it up today. We're going to complete and revise it. We have finished this session, but this is just like a revision session. Like the end of the unit, we have a review unit. So we will be going through simple revision. We might be thinking of these critical questions: that what are enzymes, what is their role, and what are carbohydrates, how can we identify them, and all these kinds of questions. We'll be going through these two videos, and after that we will have a small revision on the GCSE part about the testing of the food. I think this is the next video. I'll open it up for this one. So let's go through the first of these videos and then we'll come to that one. I think I didn't turn on the voice. Yes, let me turn on the voice and let me admit the voice and let's start.
Condensation. Hi guys, in this video we'll be looking at the biochemical basis of life, biological molecules, monomers and polymers, condensation and hydrolysis reactions, and then we'll finish with a summary.
So we don't need to look far to realise that in the natural world there is such a huge variety of life. Even just looking at the animal kingdom alone, we can see things like insects, fish, mammals, dogs, reptiles, all sorts of different animals pop into our heads. And then of course we've got other kingdoms, like we've got the plant kingdom, we've got fungi like mushrooms, and we've got things like bacteria as well. And when you consider all of the life, there's so much variety in their size, shape, colour, functions, reactions, etc., that you think that they're all built up of lots of complex differences. But despite all of this enormous diversity in the life on Earth, every organism depends on the same biochemical basis of life, and we call this carbon chemistry. So even though there is such a massive diversity in all the organisms, they all boil down to the same kind of molecules, and we call the study of these molecules that give rise to life biochemistry, and therefore there's a similar biochemical basis of life. And it all centres around an element known as carbon, which has particular properties allowing life to exist. The common ancestor that gave rise to all of us uses this carbon chemistry, and because all life is descended from this, despite all of our diversity, we all base ourselves around carbon. So down here we would have seen the last common ancestor, which is currently unknown. As it evolved into various species, of course through time, different branches branched off into different types of organisms and different kingdoms as well. For example, on one extreme we have the archaea, which are a specific type of organisms which can withstand very tough environments. We've got animals and plants, commonly called eukaryotes, and we have bacteria too. And while there are significant differences between all of these, they all rely on carbon chemistry. So what makes living things different from non-living things are particular types of molecule based around carbon and the important properties that it has as an element. So carbon as an atom exists with the ability to form 4 bonds. So carbon as an atom has the ability to make a bond with 4 other structures, or 4 other atoms. They can be different groups or the same group, but the ability of this means that the carbon atoms can bond into lots of different things and bond together to form more complex molecules. And you can imagine that if they keep forming different bonds, each of them being allowed to bond to 4 things, then the ability to make a complex molecule, which could be very large, is very high. So we can make lots of different sizes of molecules based on carbon, and it can join to lots of different chemical groups. And actually what we find is that carbon, as a skeleton like this, acts as a backbone for organic or carbon-containing molecules, and these are found in all living organisms. So the carbon chemistry is based on the fact that carbon can bind to lots of different things, and when it does, it acts as the backbone for the organic groups. And organic always refers to carbon containing. So when you think of inorganic, we're thinking of ions or things like zinc, copper, water, things which don't have carbon in them. When we think of organic, we're thinking of things like sugars and proteins, which we'll talk about next. So the biological molecules which make up life are based on carbon, as we just said. And there are four main types of biological molecule found in all organisms, regardless of where they belong in that family tree. And those four types are carbohydrates, sometimes referred to as sugars, lipids, sometimes referred to as fats, we have proteins, and we have nucleic acids, like DNA. So these are the four groups, and within these groups there are lots of different types. For example, there are many types of proteins which build up the body. There are many types of sugars that we can find in our food and in the body too. And all of them are based on carbon, but some of them have key features where they contain a small number of other chemical elements. So let's take each one in turn. Carbohydrates, you can work out what they contain from their name. They contain carbon, hydrogen, and oxygen. And if you look at the name, we've got carbo for carbon, hydra for hydrogen, and eights usually refer to oxygen. For example, when you see something like sulfate, it's sulfur bounded to oxygen. So when you look at this is an example of a carbohydrate. And let's look for these various elements. We can see carbon various times, hydrogen in lots of different places, and it contains oxygen too. Lipids contain the same elements as carbohydrates, so that's carbon, hydrogen, and oxygen, but it just is arranged in a different way. So we can still see that we have carbons, usually in a long chain, with hydrogen atoms and oxygens normally at the end. So they're the same elements as carbohydrates, but a very different structure overall. Proteins can have more than this. They have carbon, hydrogen, and oxygen, but they also contain nitrogen in an amino group, and they also can contain sulphur for some particular types of amino acid. So here's an example of an amino acid, and we'll talk about what amino acids are in just a moment. And it's part of a protein. And we can see that we've got this carbon element again with hydrogens and oxygens, but we also have a nitrogen, and we have sometimes this sulfur group, which isn't always present, but when it is, it's usually in a very low number. And then finally, looking at nucleic acids, which is that fourth group, they contain carbon, hydrogen, and oxygen as before. They contain nitrogen like protein does, and it also contains phosphorus, which is another non-metal element. So looking at this, it's quite a complicated molecule, but we can see carbon again forming that nice backbone through the structure. We've got hydrogens and we've got oxygens. We've got a few nitrogens this time, usually bound in some sort of ring structure. And we've also got a phosphorus atom, usually just the one, in a nucleotide. So as you can see, all of these biological molecules have the same feature in common. There's a carbon skeleton forming the kind of backbone or foundation of the molecule. And they usually have hydrogen and oxygen around them too, but then other elements sort of wedged in there. They often contain lots of atoms, compared to something like a water molecule which just has H2O, 3 atoms. But these tend to have many tens of atoms, sometimes more. So we often call them not just molecules, but macromolecules, with the macro meaning large, molecule meaning a group of atoms bound together. So in order to understand how biological molecules assemble themselves, we have to understand the concept and difference between monomers and polymers. So a lot of biological macromolecules don't just exist on their own, but they exist as what we call polymers. And a polymer is something which is built up from lots and lots of repeating smaller building blocks, and those building blocks individually are called monomers. So let's show an example of this. Monomers are individual molecules making up a polymer. You can see by the name we've got mono, meaning one, so that's one single unit, polymer meaning many. So lots of these one units join together to make a many unit. So just to illustrate that using a kind of Lego brick idea here, we've got 3 monomers, monomer number 1, monomer number 2, number 3, and if you add them all together, you get this chain, and you stick them together into one single polymer. And this is how most biological molecules exist.
Anybody who has any questions so far, you can raise your hand. I can explain it to you. Because this would be the last session, we are wrapping it up. And if there's any confusion, this would be the time to discuss. All right, so just raise your hand or send me the question through the chat. We'll be discussing the answers. So Waleed is raising a hand. Yes, Waleed.
Mr. I also understand this how plastic and like this
So,
send me
sorry, can you repeat that question again?
the monomer and the polymer.
Yes.
I didn't understand it well.
The monomer means like a building block.
I know the monomer is one.
Yes, it's one. And,
Yes.
and polymer means many.
And the
So,
polymer many.
yes, polymer means many blocks. Monomer means one block. So you can have one block for every type. It could be a carbohydrate block, it could be a protein block, or it could be the lipids block. So let's imagine this is the block for the carbohydrates, which we call glucose. So this red one is a glucose molecule. This is one glucose, this is two glucose, and this is the three one. So once they start joining together, they will make a long chain, and we will call it a polymer, and it will be known as starch. Starch. You understand now? How did the starch become? They are being
Yes.
produced by the same monomers, and monomer means one block, and polymer means many blocks. Sugar, glucose, is united together to make starch. Amino acids, they unite together to make proteins. And the lipids, we have the fatty acids, they unite together to make fats and lipids. Right?
Right.
Good question. So let's go to the next question. We have Mohammed, you have the mic.
Hello, Mister.
Yes, hello.
Mister, why did you say that today is going to be the last lesson?
For the biological molecules,
Okay.
for the topic biological molecules. All right. Sorry for that confusion. So sorry. Let's move on. So I'm saying that we will be moving on to the next topics. So we'll be wrapping this up.
And it's this concept of building up larger chains of molecules which help us understand how plants can get so tall, how are their stems so long, how are our bones so long. They're not made of individual units, they're made of lots and lots of units joined up into massive structures that allow us to be as big as we are. So by definition, a polymer is a long chain composed of many individual monomers which have been bonded together in a repeating pattern. And the point is, it is a repeating pattern because usually there's some sort of structure of the units following a sequence, or it can be the same unit one after another in a massive chain. So for example, in carbohydrates, the monomers are known as monosaccharides and the polymers are known as polysaccharides. So the saccharide part refers to sugar, and we either have one on its own as mono, which is the individual unit, and then when they're joined together in a chain, we have a polymer, and this would be a polysaccharide. So you'll notice that with biological molecules, the mono and the poly bit stays the same. The actual name of the bit after that is what defines it into which group. So saccharide is referring to carbohydrates. When we're talking about proteins, it's slightly different. The monomers, we call them amino acids. And then the polymers, when lots of these amino acids join together, are called polypeptides. So it's not as simple as carbohydrates this time, but we have one unit as being an amino acid, and there are lots of types of amino acids. And remember, these contain the elements of carbon, hydrogen, oxygen, nitrogen, and sometimes sulphur. And then as we join these up into long chains, we form one long polypeptide. You can kind of think of it as a peptide as an amino acid, so poly meaning lots of them. In nucleic acids, we have the monomers known as nucleotides, and polymers are polynucleotides. Sometimes you will see it as mononucleotides as well. So here's a string of nucleotides as a polynucleotide, and one of these single units would be a nucleotide. So hopefully you can see the similarities between these groups as we go through. The only one that's slightly different are the lipids. They're still macromolecules built on carbon as we said before, but they're not really classed as polymers. They don't have smaller repeating monomers. They may have an extensive length like the rest of the molecules, and they may extend much further than proteins, carbohydrates, and nucleic acids. But they're not made up of units that repeat themselves. They're simply a very long chain. So actually what they're made of instead are a number of different base units joined together in a non-repeating pattern. So the general structure that we tend to see is a molecule known as glycerol, and then when glycerol is bound to these units, these tend to be identical to each other, and these are known as fatty acids. And altogether, the structure of glycerol bound to 3 fatty acid makes a triglyceride. So now that we've gone through what the difference is between monomers and polymers, we have to understand how they're formed. It's really important because during the survival and the lifetime of an organism, things need to get broken down and some things need to be built up. For example, when we digest our food, we need to break down polymers into smaller units so that we can send them around the body and use them. If we're building our bones, if we're growing through childhood, or a plant needs to reach the sun, we need to build up larger molecules from smaller molecules. So we need ways and we need to understand how we go from one to the other and back again. So we've said that polymers are formed from monomers, and this is done by what we call a condensation reaction. So there'll be 2 types of reaction we need to talk about, one where we build and one where we destroy. So when monomers come together and they form a polymer, we call this condensation. And a condensation reaction has particular features. In a condensation reaction, there is a bonding of one monomer to another. Whether that other monomer is on its own or it's the end of a chain doesn't matter. So a monomer is bonding on to either an existing chain or another monomer. And when this happens, not only does the bond form, but a molecule of water is formed in the process too. So let's have a look at this. We've got monomer 1 and monomer 2, and they're coming together. And in the coming together, they make a polymer. And in this case, we've got 2. So we're going to call it a dimer. Now we can have monomer, dimer, meaning 2. We can have trimer, meaning 3. And eventually it becomes so many that we just call it a polymer. But there are prefixes you can put before, whereas di means 2. So now we have a dimer, we have a bond formed, and also in the process, we formed a molecule of H2O, or water, which then leaves the monomers. How does the water get formed? Well, you need to look at the groups in the monomer. Whatever monomer we're talking about, whether this be an amino acid or a monosaccharide or whatever, there's always going to be the coming together of 2 groups. There's going to be the OH of one monomer and an H of another monomer. And this is where the water comes. It's called a hydroxyl group, which is an OH from one monomer, and a hydrogen just on its own as an H from another monomer. So as these 2 come together, they form their bond. But in doing that bond formation, they have to lose these groups because they're in the way. So these snip off and form a water molecule. So you get your dimer which is formed and H2O. So it's the OH of one monomer and the H of another. And you'll find that any monosaccharide or amino acid or individual nucleotides will always have OH and H groups. And looking back on those carbon skeletons of those molecules, you can spot these quite easily. So by definition, a condensation reaction is a reaction that occurs when 2 molecules combine to form a more complex molecule with the removal of water. And that can be a monomer joining a chain or just 2 monomers binding together. So that's how we make polymers. What about breaking them down? For example, if we're digesting foods. Polymers are broken down into individual monomers by hydrolysis reactions. So this is going backwards now. This is the reverse of condensation. So here we have a polymer breaking down into individual monomers, and this is known as hydrolysis. Hydrolysis breaking it down, condensation would be forming it. And in this respect, we can talk about it as the reverse of condensation. Hydrolysis reactions require water, so we have to put water in order to break the bond between the monomers. So here we have a dimer formed of 2 monomers and a bond. In order to break that bond, a water molecule is added and it goes into the bond and pulls the monomers apart. Because what the water is now doing is replacing these 2 groups, where one monomer has that hydroxyl group and the other monomer is given a hydrogen. And you can see that water can provide these because we've got 2 H's and an O. And then when these groups form, the bond can no longer form because they're sort of in the way. The chemistry is different. So now the monomers separate and we're left with 2 monomers. So you can see how it's the direct reverse of condensation, where in the condensation reaction, we removed water to make that bond. So by definition, a hydrolysis reaction is a reaction occurring when larger molecules are broken down into smaller molecules with the addition of water. So the condensation and hydrolysis.
We have a question by Abdullah. Abdullah Iwari is asking that OH plus H equals to H2O? Yes. You can write H plus OH equals to H2O. You can write it both ways, backward and forward. In the end, it's going to be the same answer. Abdurrahman is raising a hand. Yes, Abdurrahman.
Yes, hello, Mister.
Yes, hello.
Mister, I only have a quick question.
Yes.
Is is sugar lipid
Sugar is
because it's fat?
carbohydrates.
Ah,
In
okay.
sugar, if you keep on eating too much sugar, then it will be stored as glycogen in the body. And if the glycogen is not utilized, that can be converted into fats.
Okay. Thank you,
All right.
Mister.
You're welcome. So let's go.
These reactions are used to build up or break down all of the biological molecules, lipids, nucleic acids, proteins, and carbohydrates. And here's a table just to summarize them. In condensation, monomers are joined together, whereas in hydrolysis, monomers are broken apart. Condensation forms water, and hydrolysis requires water. If you have trouble remembering which is which, remember that condensation is what you see on the sort of mirror in the bathroom or on a cold bottle, where water starts forming in droplets. So it's forming water. And hydrolysis, well, lysis means to break. So in this case, we're adding water and we're breaking it into two to make those groups again. And in condensation, we form a bond, whereas hydrolysis, we're breaking. Again, breaking, break, lysis. And condensation and hydrolysis reactions are very important reactions taking place in all the cells of organisms. So they're examples of metabolic reactions making useful products.
Hey guys, I hope you enjoyed the video. If you are looking for an amazing A-level biology resource, join me today in my series.
All right, that would be for this video. The second video is about testing the, we'll come back to this one again. This is a more simpler version of the same topic. So let's go through this one as well. And after that, we'll go with the testing of the food, if the starch is present, if the carbohydrates, by choosing the biuret, benedict, and the iodine potassium iodide solution.
Go ahead and think for a moment about your very favorite food. What is it? Pizza, macaroni and cheese, chicken salad, sushi? Well, we all have different food preferences, but food is a source of large molecules that are needed for life called biomolecules. There are 4 major biomolecules that make up all of life, and this will be the focus of this video. Before we get into details about the 4 biomolecules, we need to talk about one very important vocabulary word, the word monomer. A monomer is a building block. If I had some large substance, the parts that make up the substance are called monomers, just like building blocks. We're going to talk a lot about monomers today because we need to understand what the biomolecules are made of. And we need to understand biomolecules because they're building components of life. So let's introduce the 4 biomolecules now and talk a little bit about their functions. We'll start with carbohydrates, carbs. Well, carbs are something that you've probably heard a lot about when people are talking about diets. You know, they try to go low carb, or maybe they want a lot of carbs. Diets always come and go. Pasta and breads are examples of foods heavy in carbohydrates. Carbs are actually a very important source of energy. In fact, that's one big function of carbs. They are a great, fast source of energy. If you are a marathon runner, you might want to eat a lot of carbs the night before a race. Lots of marathon runners do this. It's called pasta loading. They eat a big pasta dinner the night before they go out on their marathon. Now, carbs have a monomer. Again, remember, monomers are building blocks. The monomer for a carb is a monosaccharide. I know that's a big mouthful, but monosaccharides make up carbohydrates. Next one up is a diverse group known as lipids. Lipids are better known as fats, and they have 2 different types of building blocks. One type of building block is called a fatty acid, and the other type is called a glycerol. Now, examples of lipids include butter, oil, and cholesterol. Lipids, though, they have a lot of great functions. You might think, well, that's fat. How good can fat be? Well, it just depends when you put it into context. For example, you know those really adorable seals that you see on calendars? They have this fluffy white hair. They're actually called a harp seal. Well, they only look like that when they're babies. When they get older, they're not quite as cute. In their little baby stage, they actually have a lot of this hair that they're born with that keeps them warm. But over time, they have to develop blubber. It's fat and it helps keep them warm. Lipids are great for insulating. Also, you might not think about fats as being related to energy, but fats are a great source of long term energy. They can store energy for a long, long time. Say, for example, you wanted to swim the English Channel. That's like 21 miles of swimming. And the fastest swimmers might be able to do that in 7 or 8 hours, but it might take a lot longer than that for the average swimmer. More like 25 hours, and that's a lot of swimming. Well, you would want to make sure that your body has enough lipids, enough stored fat that it can pull upon. Because after you burn off those carbohydrates, remember, carbs are the fast source of energy, well, you might not have enough energy storage unless you have some lipids on hand. Lipids also make up cell membranes, so they are very important for life because all living things are made of cells. Of course, an excessive amount of lipids could be a bad thing for your health. Remember, it's all about moderation. Okay, next, proteins. Now, when you hear about proteins, a lot of times you might think about power bars. They say they have a lot of protein in them and that they help with muscle building. Well, protein is great for muscle building. Examples of foods that are high in protein include meats and many types of beans. The monomers of protein are amino acids. So sometimes you see these labels that say, this has 20 amino acids in this food. And really, they're just trying to say that it has protein. Because proteins are made of amino acids, so that's just some fancy advertising for you. But in addition to being important for muscle development, protein is also very important for other functions, such as working in the immune system and acting as enzymes. Remember, enzymes are made of proteins, so proteins are important for the body. Now, when we start talking about genes, the DNA genes, not the genes you wear, the DNA codes for proteins that are very important for structure and function in the body. The last biomolecule is known as a nucleic acid. Nucleic acids include DNA and RNA, which we'll get to when we get to genetics. They have a monomer called a nucleotide. That's going to be an easy one for you to remember because nucleotide sounds a lot like nucleic acid. And if considering DNA and RNA, both of these are involved in genetic information for the coding of your traits. They are found in a lot of your food because whenever you eat something that came from something once living, it can still contain the DNA. For example, when you eat a strawberry, you're actually consuming all the cells that make up that strawberry. And in the nucleus of all those strawberry cells is DNA. Plants and animals both have DNA. In fact, any type of life must contain nucleic acids to direct the cell's activities. So we just powered through introducing the 4 biomolecules by providing examples, exploring.
So we have a question that what is an RNA? Waleed, the DNA contains the information, right? And there's another nucleotide which is responsible to follow these instructions. So the one who follows these instructions is the RNA. They are exactly the same. The DNA and RNA looks exactly the same, except for one thing, that RNA contains oxygen, DNA is without an oxygen atom. So if you're looking into the monomers, you're going to find DNA is without oxygen, RNA is with oxygen, and DNA is only the information, whereas RNA is actually the doer. You know, understand by doer, the one who does everything. He reads the instruction from the DNA and does everything as it is being told. So RNA is the performer. He is the manager who follows the instructions from the DNA, who gets the job done. We will have a complete session on this about the RNAs. There are 3 different types of RNAs. So we will be reading there's a complete lesson on it, inshallah. Okay, I hope this answers the question. Let's go back.
Their monomers and giving some general functions. One last very important part to mention is the structure of these biomolecules. Understanding the structure can help with predicting their properties and easily being able to identify them. One thing I like to tell students to do is to write the 4 biomolecules in the same order that we went through: carbs, lipids, proteins, and nucleic acids. Then remember this mnemonic device that goes with these 4 biomolecules: cho, cho, chon, chomp. Instead of chomp at the end with an M, it's chomp at the end with an N. The C stands for carbon, the H stands for hydrogen, the O for oxygen. So carbs, lipids, proteins, and nucleic acids all have that cho in there. It's just that proteins and nucleic acids also have an N, which is nitrogen. And nucleic acids additionally have a P, which is for phosphorus. So again, cho, cho, chon, chomp, the major elements in the 4 biomolecules. Now, these elements are arranged differently in the 4 biomolecules. It's important to explore the arrangement of the elements in biomolecules because the structure of that arrangement greatly impacts the biomolecule function. So, to the Google to discover some biomolecule arrangement illustrations. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so we'll go to the one last session. It's about the finding out the protein or carbohydrates or lipids. Let's see the test. Remember the terms biuret, Benedict, and the iodine solution. We use the iodine solution for starch, Benedict for glucose, and biuret for protein.
A cheeseburger contains a juicy beef burger, oozy cheese, crisp lettuce and tomato, and sweet tomato ketchup, all held in a soft bread bun. No, this isn't an advert for fast food or a way of making you hungry. We're going to see if a cheeseburger contains some of the main food groups: starch, sugars, protein, fat, and vitamins. How? By discussing food tests.
Let's start with starch. Starch is a carbohydrate made up of long chains of glucose molecules. It's a food store in plants. Which part of the cheeseburger do you think will be high in starch? It's the bun, because buns are made from flour, which contains starch from wheat.
To test for starch, you use iodine solution, which is an orange-brown colour. When iodine solution is added to starch, it forms a complex with starch molecules and goes a blue-black colour.
What happens when iodine is added to the bun? It goes blue-black. This is a positive result, so the bun must contain starch. What about sugars, or more specifically, a type of sugar called reducing sugar? Glucose and fructose are reducing sugars found in plant material. The tomato ketchup is sweet, so it's likely to contain sugars. How do you test for reducing sugars?
You take a small amount of the food and mix it with Benedict solution. If the food is solid, you'll have to grind it up with water in a pestle and mortar, then add it to the Benedict solution. You then heat the mixture in a water bath. Benedict solution is blue but turns green to orange and then brick red if reducing sugars are present. The tomato ketchup gives a positive result, so it does contain a reducing sugar.
Next up, what about proteins? What parts of the cheeseburger might contain protein? Well, the burger and the cheese are both animal products. Let's test the burger. First, because it's solid, you need to grind it up with some water to form a liquid. Then you add some of the liquid to some biuret solution and wait for a few minutes. If protein is present, the pale blue colour of the biuret solution turns purple.
Yes, another positive result, so the burger contains protein. Cheese is high in fat. To test for fats and oils, you use the ethanol test. First, you grind up the food with some water. Then you add some of the food mixture to a test tube containing ethanol. The cheese mixture turns the clear ethanol cloudy, and this is a positive test for fat.
So we know that the cheeseburger contains starch, sugars, protein and fat. What else? Let's test for vitamin C. You might not think a cheeseburger contains any vitamins, but the tomato is a fruit, so it may well contain some vitamin C.
A chemical called DC-PIP is used to test for the presence of vitamin C. The first step is to grind up the tomato to extract some juice. You then add drops of blue DC-PIP one at a time and shake the mixture after each addition. Count the number of drops as you go. To begin with, the blue colour of each DC-PIP drop disappears. But after you've added several drops, the mixture stays blue. Record how many drops you added before it stayed blue. The more drops you added, the more vitamin C the food sample contains.
But beware, if the food you're testing has a strong colour or more than 5 centimetres cubed of DC-PIP is decolorised, you need to dilute the food mixture. You can use this test to compare the amounts of vitamin C in different foods by using a burette to measure the volume of DC-PIP needed to decolorise the foods. The test shows that the tomato is quite high in vitamin C content. So our cheeseburger contains all the main food groups: starch, sugars, protein, fat and vitamin C. At least one part is good for you.
All right, that would be all for today. And some people are asking that if the tomato is a fruit or a vegetable. Yes, tomato is a fruit. We all have been misconceived about the tomato, that it's a vegetable or it's not a vegetable, it's a fruit. Yes, you can Google, and yes, it is a fruit. So I'll be stopping the recording now.